Hello everyone. Today's topic is incomplete dominance. So if we see Mendel first law or Mendel law of dominance, if we see Mendel's law of dominance, what it says. Mendel law of dominance states that in case if there are two pair of contrasting characters, okay, suppose this T is for tallness and small t is for dwarfness. So if there are two pair of contrasting characters, then only one, that is dominant, express itself. It itself. Okay. So suppose this T is dominant. If we say this capital T is dominant and the small t as recessive, then when they will come together like capital T and small t, the phenotype that develops will be tall. Okay, so this is the law of dominance, that when a pair of contrasting characters they come together, only one of them is expressed. The one that is dominant will express itself in the F1 generation. But after Mendel, there are certain other examples where such a trend is not followed. For example, uh, in case of flower color in Snapdragon, that is also called as dog plant. Let's see this case here. Two kind of flower colors are there. What what flower color? Now see, a red flower, hmm? Red flower, hmm? And the other one is white flower. Okay? White. One is red and the other is white. Now the red as genotype capital R, capital R. It was considered dominant. Hmm? And the white has small r, small r. This is the genotype. Now if we see gamete, gametes, the red flower will yield only one kind of allele, that is capital R, and the white flower will give allele with a small r. Now in the F1 generation, what is obtained? Capital R and a small r. And it was found that just that the phenotype of this genotype has neither red color nor white color. Rather, they yielded an intermediate color that was pink. Which color? The color was, it yielded a pink color flower. Hmm? So what was this? Since neither red was dominant nor white was dominant, hmm, then the color which was obtained was pink. So it was said that this is incomplete dominance. Hmm? Now, if we cross, now let's see in F2 generation. For F2 generation, if the F1 capital R small r was crossed with capital R small r, hmm, so what would we get? This is a unit square. In F2 generation, the gametes will be capital R. For this generation, the gamete would be capital R, small r, and for other capital R, small r. So the genotype is capital R, capital R, capital R, small r, capital R, small r, and small r, small r. So in F2 generation, what was obtained? We can see phenotype. This is red, pink, pink, and white. So types of genotype: red is to pink is to white. Hmm? How many red? We got one red, two pink, and one was white. So one red, two pink, and one white. And so what was this? Was genotype. And if we talk about phenotype, the phenotypic ratio was also the same, 1 is to 2 is to 1. So in case of incomplete dominance, genotypic typic ratio is equal to phenotypic ratio. Okay, I hope it is clear. Now let's see another thing. What was the reason, hmm, for concept of dominance? Okay, okay. Now in a genotype, huh, one genotype is represented by, hmm, a pair of allele. Suppose this is one allele and this is another allele. Hmm? So a pair of alleles, that is a pair of alleles, hmm, is represented by pair of alleles. So allele number 1 and allele number 2. Hmm? And it may be capital R or capital R, capital R, or small r. So in all the case, a pair of alleles are present. Now, why a phenotype is shown by a particular genotype? Now, let me explain to you. This is the genotype. This genotype is represented by a gene. Hmm? Okay, and the gene show their effect by translation of functional protein. Hmm? If the functional protein is there, we will see the particular phenotype. And if a particular gene is non-functional or mutated, in that case the phenotype may be non-functional phenotype, or if mutated, then we can see altered phenotype. Okay, now mm, let us consider three cases. Hmm? Let us consider three cases that are possible. Now the first case is the other allele, hmm, or the modified allele produces normal or less efficient protein. Okay, the second case, the modified allele produces a non-functional protein, and the third case, the modified allele produces no protein at all. Okay, mm, so here the modified allele is similar to the unmodified allele. Hmm? Hmm? So what will happen यहाँ पे, same phenotype will be produced. Hmm? it codes for a non-functional protein, then the non-functional protein, uh, it means non-functional protein and no enzyme. Hmm? In both the cases, the phenotype will be affected. In both the cases, in case of non-functional protein, or there is no protein production by that modified allele, in both the cases the phenotype getting affected. ठीक है? Now see, if the modified allele produces protein that is equivalent to either normal or less efficient protein, then the same phenotype will be produced. Okay, but so it is this will be dominant. Dominant allele. But if the modified allele produces either a non-functional protein or no protein at all, hmm? So these both cases are as good as recessive allele. Now let's see another case where the modified allele, what it does, it produces a functional, okay, it produces a functional but an altered product. It produces a functional, it is a product of the modified allele which produces a functional product. In that case, this case will be treated as a case of incomplete dominance. Okay, it will be treated as a case of incomplete dominance.